So students, today I will be explaining about concept called pleotrophy, multiple allelism, and polygenic inheritance. And what are the main points in these concepts? So I'll start with pleotrophy. Pleotrophy is expression of many characters by a single gene. It means that there is a single gene and it is going to express many characters. This phenomena is called as pleotrophy. But this gene is called as pleiotropic gene. Okay, so the pleotrophy is a phenomena where a single gene expresses many characters, and the gene involved is called pleotrophic gene. Now, best example which is given in pleotrophy is phenylketonuria and even sickle cell anemia. Like this. These are monogenic disorders. These disorders are one which will occur due to mutation in a single gene. Okay. So these all are also called as Mendelian disorders. So monogenic disorders or Mendelian disorders, and it is due to mutation in a single gene. Now in this disease, there are many symptoms appearing. Now, if I take example of phenylketonuria, this disease is caused by defect in a single gene that encodes for an enzyme called phenylalanine hydroxylase. So that enzyme becomes defective. When this enzyme becomes defective, the phenylalanine accumulation occurs, and that is going to convert into certain derivatives like phenylalanine, phenyl lactic acid, and phenyl pyruvic acid. These derivatives are harmful and they pass through blood circulation and they may cause mental retardation. And therefore, one of the symptoms of phenylketonuria is mental retardation. So I'll begin with an example: phenylketonuria. So this is about one of the symptoms of phenylketonuria. Apart from this, there is a change in the coloration of skin. The skin becomes light. Why the skin becomes light is because of less concentration of tyrosine, and that tyrosine is not going to get converted into, when tyrosine is not there, melanin cannot be produced. As a result, the skin is going to be light skin, and wherever there is a melanin content, there is a lightness. That means there is no melanin pigment, it becomes light in color. It means that this single disease is manifesting itself into different characters, okay, and into different symptoms here, and that is why we say it is a pleotrophic in nature. This monogenic disorder, this genetic disorder is a pleotrophic, exhibit pleotrophy, and that gene is called pleotrophic. And the next one will be what is called multiple allelism. Already multiple allelism has been taught by the beginning of this week itself. And now I will just elaborate, I will explain further, has to only explain the important points in multiple allelism. Multiple allelism is when a gene exists in more than two allelic forms. Okay, that means if the gene form is more than two, it means. For example, best example of multiple allelism is human blood group. Human blood group is best example of multiple allelism. So here, gene for human blood group is given by an alphabet called I. So I stands for gene for the human blood group. Now what are the alleles of this gene? What are the alleles of this particular gene? So there are three types of alleles of this particular gene. As I already told, that I stands for isoagglutinogen. So here alleles is three types are there. One is IA, another is IB, another is IO. Instead of IO, you can also write small I in order to indicate that it is a recessive allele. So as you can see, two of the alleles are dominant, and one of the allele is recessive. And based on this, there can be 6 phenotypes in human blood group, and there can be 4 phenotypes in human blood group and 6 genotypes. So number of phenotypes in human blood group is 4, whereas number of genotypes in human blood group is 6. Now 4 is for A blood group, B blood group, O blood group, and AB blood. Whereas genotypes will be for homozygous A, heterozygous A, then homozygous B, heterozygous B, and AB blood group, and followed last is the O blood group. In this, it has been explained in codominance that this AB blood group actually is an example of codominance. AB blood group is an example of codominance. So these are the different phenotypes as well as genotypes. Genotypes means allele combinations called genotypes. So this was what is called multiple allelism. The definition actually falls here. You can see how many alleles are there, more than 2. 1, 2, 3. More than 2 if alleles are there, then we call it as multiple allelism. Okay, so this is about pleotrophy and multiple allelism. Now, blood group, I shall just show one of the blood group problems. Suppose If there is an individual who is heterozygous for A blood group and a person will be of heterozygous B blood group, that means both the male is heterozygous A, female is heterozygous B. Then what will be the combination of blood groups in the offspring? So you know that two gametes can be formed from this IA and I. Similarly, here also two gametes can be formed IB and I. So offsprings produced from these possibilities will be, so one is IA and I, another is IB and small I. And if it is felt, one possible blood group is AB blood group, another possible blood group is heterozygous B blood group, another possible blood group is heterozygous A blood group, another possible blood group, the last one is O blood group. So if both the parents are heterozygous for the blood group, then offspring will have all the blood groups. So offspring can contain all the blood groups. For example, this is AB blood group, this is, so AB is possible, B blood group, A blood group as well as O blood group. All the blood groups are possible when the parent are heterozygous. This blood group, human blood group is determined on presence of antigens on the surface of RBCs. These antigens are supposed to be located on the cell membrane of RBC. Now antigens can be A type and B antigen. Okay, and they will be expressed by IA and IB. Now these antigens itself will be located on the surface of RBCs. These antigens are glycoproteins. Which can be divided into, glycoproteins means it can be divided into the carbohydrate part and the protein part. So it's conjugated with carbohydrate. So this is carbohydrate and protein part. Such type of glycoproteins is the chemical nature of antigens present. If the person's RBC is coated with A antigen, then his blood group will be A blood group. If the RBC is coated with B antigen, then a person's blood group will be B blood group. And if the person's RBC have both antigen and antigen A as well as antigen B, then the person's blood group will be AB. And if the person do not contain any of the antigen, then his blood group will be when it comes to blood group, you should know what is universal donor and universal acceptor. Universal donor is O blood group. O blood group is a universal donor, whereas AB blood group is a universal recipient. Is a universal recipient. O blood group person can donate the blood to almost all of the blood groups, that's why universal donor, and A blood group can accept all the other blood groups, so it's called as universal recipient. Then universal donor, why? Because there is an absence of antigen, and here why? Because there is an absence of antibody. Absence of antigen will not trigger the blood transfusion reaction. There is no immune reaction there. So absence of antibody also, there cannot be immune reaction. Here, absence of antigen will not trigger an immune system in the recipient. Here, absence of antibody in the recipient also will not trigger. So basically, a recipient is a recipient should be the main subject whenever it comes to blood transfusion, because immune response is exhibited by recipient and not the donor. Then, other blood groups, if it is A blood group, it can accept blood from A blood group as well as O blood group. If it is B blood group, it can accept the blood A, B as well as O. And if it is AB, it can accept the blood from AB and O. And O blood group can accept the blood from O blood group only, whereas it cannot accept from all the others. So this here you can see universal recipient, and this will be universal donor. So this was about how the blood transfusion can occur from donor to recipient. The next topic is polygenes. Polygenic inheritance. What do you mean by polygenic inheritance is if many genes encode for the same single character, then that group of genes are called polygenes, and the inheritance is called polygenic inheritance. Now consider if there is gene 1, say gene 2 and gene 3, these all genes together will express a single character. When many genes express a single character, then it is called polygenic inheritance. If you consider here, human skin color is a polygenic inheritance. Human skin color is example of polygenic inheritance. So basically, human skin color is not determined by a single gene, but it is determined by 3 genes. Okay, they are represented as A, B, C. Polygenic inheritance in humans was studied by Davenport. And the list of genes responsible is 3 of them. So genes responsible for polygenic inheritance is A, B, and C. These are the genes, representations of the genes. And each gene exists in 2 forms. Those are called as allelic forms, which will be dominant, recessive. Here also B exists in 2 forms, dominant and recessive. C gene also exists in 2 forms, dominant and recessive. So therefore, if 3 genes are there, they exist in 3 allelic forms. So this is about the genes involved in inheritance of skin color in humans. So, what is the cross performed in polygenic inheritance? I told you who described polygenic inheritance is Davenport studied this polygenic inheritance in humans skin color. And what do the cross involve? The cross of polygenic inheritance involve the individual who has very dark skin. The individual with dark skin will be crossed with an individual who has light skin color. So this is basically what you call the Negroes as well as white Caucasian Negroes and Caucasian crosseters. So this is person who is Caucasian. Here is they are going to have a white color, and that too it should be very dark, and they should be very white. Got? So this is the parental generation in this polygenic inheritance. These two are parental generation. One is very dark skinned individual and another individual is very white skinned individual. Therefore comes Negroid as well as Caucasian crosseters. So what are the gametes produced by one of the parent? That is very dark parent. So gametes should represent every gene. Okay. So this is one gene. If it is diploid state, its haploid should come in the gamete. This is another gene. This is double diploid state. So haploid condition should come in a gamete. This also same thing. So only one type of gamete is produced. Similarly, the second parent also produces only one type of gamete because all are in homozygous condition. So therefore, the gamete for the second parent will be A, B, C. In F1 generation offspring, the intermediate phenotype is obtained. Okay. And what is that is, the genotype therefore will be union between the gametes to produce F1 generation. And the genotype of F1 generation is heterozygous. Heterozygous at all the three locus. Understand? And this individual has, it does not, the individual of F1 generation do not either is very dark or he is not very white. He is intermediate. Understand? So this intermediate individual is named as mulatto. Intermediate individual is named as mulatto. Mulattoes will be intermediate between characters of the very white and very dark. So they are heterozygous and they are named as mulattoes. Now crossing with the same genotype. If the same genotype is considered here, in animals will be called interbreeding. Then this will result in F2 generation. Okay. So if the F2 generation table is drawn here, so total of 8 gametes are produced by each parent. So consider this one. This will be on one side the male gamete, the other side the female gamete, like this. So 8 gametes will be produced. What are the 8 gametes is, one will be A, B, C. One of the gametes will be A, B and C. Another gamete is going to be A, capital B, small c. Another will be capital A, small b, big C, capital A, small b, capital C. Then capital A, small b, small c. And now another 4 gametes will be beginning with recessive A. So small a, capital B, capital C. Small a, capital B, small c. Then small a, capital B, small b, excuse me. Capital. And finally, all are small s. So you can see a total of 4 gametes have been formed. Now these gametes has to be filled in the male and the female. So in the male, you will write the first gamete like this, and then comes the second gamete, third one, fourth one. The next one is these are the 8 gametes that has to be entered in order to know what are the different phenotypic characters in the F2 generation. So again here also it will be ABC, 8 different gametes has to be written here. So here also total 8 gametes. So this is the Punnett square for 8 gametes. So male 8 gametes, female 8 gametes. Therefore, the total number of offsprings will be 64. So this is the Punnett square for the polygenic inheritance. Now I will tell what is the phenotype which has to be written here. Whenever all are dominant genes, the phenotype will be very dark. Phenotype is going to be very dark. Phenotype is going to be dark, understand? Very dark. And second one. Now here, one of the allele is becoming recessive. When one allele is becoming recessive, the person's phenotype will be just dark. Then next, like this, it has to be red. So in this allele combination also, there is one recessive and the remaining are dominant. That means 5 alleles are dominant. When 5 alleles are dominant, again it will be dark. Understand? Now next is A. Now 2 alleles are what is called 2 alleles are recessive and 4 alleles are dominant. In that case, it will be fairly dark. Understand? Now the next one here, you can see only one is recessive. It means again it is only dark. What? Here, 2 are recessive, remaining are dominant. Again, it is fairly dark. The next one. Here also 2 are recessive, so it should be fairly dark. And here will be heterozygous condition. Here it is going to be heterozygous condition. So this is a mulatto, that is intermediate. Mulatto. Okay. So intermediate character is the mulatto. So this is a mulatto. But so now further this now next will be the characters is going to change. For example, let me go few of the cells and then I'll explain what needs to be explained. So A. So this person is going to be dark. This person also will be fairly. So this is going to be a mulatto, intermediate phenotype, mulatto. Intermediate phenotype. This will be again, what is that? 2 recessive and remaining will be dominant. Okay. So it is going to be fairly dark. So in a similar way, the offspring has to be written. Now how the offspring has to be identified? How the phenotype of offspring of these has to be identified? In the same way, it has to be filled in all the cells. So I have shown few of the ways in which you have to write the genotype and further identify the phenotype. So what I shall do is, like this, I have shown some procedure how you have to fill the Punnett square. Later on, you have to identify the phenotype. Next is how to identify the phenotype in polygenic inheritance. So identification of phenotypes. So phenotypes in polygenic inheritance. Okay. So what are the different phenotypes? How to identify? Depends on number of dominant. Depends on the number of dominant genes, dominant alleles. Let me tell, not genes, dominant alleles. Now I shall give example. If in that F2 generation Punnett square, if you want to identify the phenotype, if suppose all of the alleles are in dominant state. So if you count the total number of alleles, all the 6 will be in dominant state, then the person's phenotype is going to be dark. So that is this phenotype. Now in case if this condition is there, where there are 5 dominant and 1 recessive, in that case, any of them, any one of it is recessive, remaining 5 are dominant, it means that it will be, excuse me, this is very dark. and this is going to be dark. So this is an example of dark phenotype. So next is if two alleles are recessive, then the individual is going to be fairly dark. Fairly dark. If three alleles are recessive, this is an intermediate phenotype, also called mulatto. Also called as mulatto. Mulatto's features, they may have some features of white and some features of negroes. Okay, but they do not exactly resemble their biographical features. So these are intermediate types. In the mulattoes, the number of alleles in recessive state is going and the dominant state, number of alleles in the dominant state, both are same. Now, if the number of alleles are only 2 dominant and 4 recessive, then the phenotypic characters will be fairly white. Fairly white. And now further, if you write the phenotype, if only one dominant allele is there, then it is called white. And if all the alleles are recessive, then the phenotype will be very white. If you observe these phenotypes, you can write any phenotype in the F2 generation Punnett square. So what you noticed is ultimately dominant gene is the one that is imparting a dark color to the skin. Okay. So more the dominant alleles, more darker is the skin. That's why we say it is a cumulative effect. So polygenic inheritance is an example of cumulative effect. Okay. And skin color of human being, skin color depends on a pigment, and that pigment name is melanin. So this pigment name is called as melanin. So, and this melanin is controlled by dominant allele. And what is the dominant allele here? Capital A, represented by capital A, capital A, capital B, as well as capital C and capital C. These are the dominant alleles. So these dominant alleles only are responsible production of what? Particular quantity of melanin, because of which the skin color is going to be darker. Melanin is basically a brown pigment. Okay. As the number of dominant alleles becomes less in a recessive state, there is like it keeps on becoming lesser and lesser as the number of recessive alleles keeps on increasing. So that is about how the phenotype should be assigned. Okay. So finally, what is the phenotypic ratio in polygenic inheritance? So I am going to write it here itself. Phenotypic ratio in polygenic inheritance is so 1:6:15:6. And these four corresponds to all these characters. Starting from here corresponds to all these characters given. So this ratio corresponds to this. Okay. So this was about polygenic inheritance. So therefore, in a very brief revise will be pleotrophy is what? It is nothing but single gene, many characters. Whereas polygyny is kind of opposite. Many gene, single character. And here, in case of multiple allelism, genes are not, it is a single monogenic only, but thing is number of alleles are more, and therefore we say it is multiple allelism.